You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast. I have Josep Bassaganya-Riera. He's the chairman and CEO of a company called Landos Biopharma. And the website is landosbiopharma.com. Josep, thank you for coming. How are you doing today?
Doing great. How about yourself? Thanks for having me.
Yeah, no problem. So tell me, what's the premise of the company? What's the premise of Landos and what are you working on?
So Landos is a company that develops safer and more effective therapeutics for autoimmune disease. There's an unmet clinical need for therapies that have a more benign safety profile, particularly in Crohn's disease and ulcerative colitis, but also other autoimmune indications. And Landos has a unique expertise in the interface of immunity and metabolism that we are harnessing to identify new therapeutic opportunities to tackle these widespread and debilitating diseases. The unmet need in Crohn's and UC is connected to the use of drugs that have significant side effects, including cancer, infection, and death. And in addition, they are generally injectables. In many cases, they require infusions in the hospital. Landos is focusing on developing not only therapies that are safer and more effective, but also more convenient for addressing those unmet needs of autoimmune disease patients. Our lead asset is BT11, an orally active, gut-restricted, small molecule therapeutic for Crohn's and UC. It targets a new mechanism of action, the Lanthionine Synthetase C-Like 2 pathway, one of those pathways in the interface of immunity and metabolism. It's a dual mechanism of action. Not only this pathway decreases the inflammatory responses connected to tissue damage and disease in Crohn's and ulcerative colitis, but also it increases anti-inflammatory responses. And the net result is that we provide mucosal healing and recovery faster with a better adverse side effect profile.
Why has it been such a challenge so far to come up with a better protocol for Crohn's disease and other IBD, et cetera? What's so difficult about it?
Yeah, I think Crohn's and ulcerative colitis, and I would say this is applicable broadly to the autoimmune disease space in general, suffers from the challenge of decreasing the immune responses, making the immune system less capable of causing damage, of inducing disease, while at the same time not turning it off too much that you may inadvertently induce immunosuppression. And the general approach that has been used for the last 15, 20 years is a very prevalent and dominant use of biologic therapies, which they are in many cases targeting one cytokine, one of the inflammatory elements that the immune system produces. For instance, TNF-alpha would be one of those examples, right? TNF-alpha blockers, when you inhibit that component of the immune system, you are only taking into account a single component in a massively and dynamically interacting network. And there's generally redundancies that are embedded within how the immune system works. So you may block that a specific cytokine, that specific molecule, but then there's other pathways that are equally inflammatory that compensate for that inhibition. As opposed to that approach, which has the side effect profile we've discussed before. We are going after a very selective novel pathway, Lanthionine Synthetase C-Like 2, that once activated, it triggers the inhibition of several of these inflammatory markers, TNF-alpha, but also interferon gamma, interleukin 6, MCP1, and more importantly, it increases the anti-inflammatory arm of the immune system. And so when those two arms work together, the decrease of the inflammatory cytokines that are responsible for the damage within the gastrointestinal tract in Crohn's and UC, and at the same time, activation of the anti-inflammatory responses connected to mucosal healing, the therapeutic efficacy is very dramatic. And in fact, in our preclinical testing, we have shown that BT11, our lead compound for Crohn's and UC, is not only efficacious in 5 mouse models of inflammatory bowel disease, but in 2 of these models, we've done head-to-head comparisons of BT11 versus other drugs, including biologics and 5-ASA, and BT11 has been able to outperform. So we're very excited to have this very robust preclinical package. We've completed phase 1 clinical testing last year, where we showed that BT11 has an outstanding safety profile. Even at high doses, the adverse event profile of BT11 was no different than placebo. And importantly, we're able to show that one of the key biomarkers of inflammatory responses was decreased by BT11. That was very encouraging and was consistent with what we had seen in our previous preclinical studies. And now in August of this year, we announced the dosing of the first patient in a phase 2 study with 195 ulcerative colitis patients with 3 groups: a low dose BT11, high dose BT11, and placebo. And we are going to conduct this study over next year, and we are excited to have the data in the second half of 2020.
Yeah, it's weird. When there's an autoimmune response, it's selective. It seems to target a certain part of the body and not the entire body. Otherwise, I'm sure the entire body would fall apart. But yet, our response to fixing that is not selective. I guess it's been to just suppress the immune system, or certain aspects of it. Have you identified why, in particular, let's say in Crohn's, that part of the body is attacked? Like, how does the immune system know? How does it know to target those particular types of cells, and why? Do you understand that mechanism?
Yeah, I think that we are a company that is basically designed with the mission of serving the unmet clinical needs of these patients. And in order to do so, we are very embedded in the science. So the mechanistic understanding of BT11 and the LANCL2 pathway or other targets that we have in our development pipeline is absolutely crucial. And you are bringing up a great point. So there's a broad range of autoimmune diseases from Crohn's and UC that are gastrointestinal tract oriented, where we have lupus that involves, the spleen, the kidneys, rheumatoid arthritis, the joints, multiple sclerosis, the nervous system, and so on and so forth. And the reality is that the prevalence of autoimmune disease is growing. So there's more people that are diagnosed with Crohn's and UC, but also other autoimmune disease in the US and worldwide. There's a variety of hypotheses that support that, from the hygiene hypothesis that the immune system in developed societies is not as challenged as in other societies, and therefore it gets out of sync and starts responding against parts of our bodies that usually it should not respond to. The immune system is designed to destroy infections, to destroy bacteria, viruses, fungi. At the same time, is designed to destroy tumors in the case that some people develop, cancer. And excessive, dysregulated, uncontrolled responses of the immune system in the context of autoimmune disease are causing significant damage, are disrupting the life of patients, are disrupting the life of those families. And we've been very systematic and deliberate in how we approach, particularly Crohn's disease and ulcerative colitis, and that's what I'm going to talk about. But we have a pipeline of other assets for other autoimmune indications, including lupus, rheumatoid arthritis, multiple sclerosis that are at preclinical stage, IND enabling stage. But getting back to BT11, our lead asset for Crohn's and UC, before we started the development program, we did an in-depth analysis of what would be the ideal drug given this disease and given the unmet need of the patients. And we realized that we wanted to develop a drug that was provided orally as a once-a-day tablet. Not 2 pills a day, but 1 pill a day. And the oral was going to be much more convenient than an injectable or an infusion in the hospital. We also realized that some of the problems in the current drugs for Crohn's and UC are the side effect profile. This cancer, infection, and death, the black box warnings connected to some of those. And so we wanted to design a drug that would act locally in the gastrointestinal tract, bring the immune system back in sync through a novel mechanism of action, the LANCL2 pathway that I've described earlier, but that would not have significant systemic exposure so that it will not cause a systemic immunosuppression. So the immune system in the rest of the body would be able to continue to fight infections, cancer, and other diseases, while in the gastrointestinal tract, where in Crohn's and ulcerative colitis, the immune system is excessively activated and out of control, our therapy would bring it down. And so BT11 had a checkbox in each of these boxes, had a check mark in each of these boxes where it's orally active, gut restricted, has limited systemic exposure. And we're very excited that in a matter of 18 months, we've gone from early conception, preclinical stage, to a phase 2 ready asset. And now we are in the middle of a very robustly powered global phase 2 clinical trial with about 50 sites around the world, 11 countries. We find our key opinion leaders are very excited about the new mechanism of action, the toxicity profile, the potential for safety. So we are excited with what we've accomplished so far about BT11. I think that it has the potential to show an outstanding safety profile in the context of impressive efficacy.
How selective is this pathway, or how unique is this? Is this pathway unique only to Crohn's, or is it various, or any condition, any autoimmune condition that affects the gut seems to have this pathway involved?
So the pathway is quite critical in immune cells. And in fact, we've created knockout mice that do not have the LANCL2 pathway, and we've demonstrated that their ability to respond to the drug is impaired. So if we compared a wild-type mouse versus an LANCL2 knockout mouse, and we give them BT11, we induce IBD, the one that expresses the pathway will show therapeutic efficacy. When the pathway is not there, the therapeutic efficacy is not present. So the drug is very selective. Now, this pathway is present in all immune cells, not only in the gastrointestinal tract. So if we have cells in the spleen, that would be relevant for lupus. This LANCL2 pathway will play a role, and we can pharmacologically activate that pathway and elicit therapeutic activity. In the joints, the same thing. But remember, for the Crohn's and UC, we designed a drug that is locally acting, that it's gut restricted. It doesn't it's not absorbed. Very small concentrations make it to circulation. So BT11 is designed to activate the LANCL2 pathway in the gastrointestinal tract and provide therapeutic efficacy in Crohn's and UC and inflammatory diseases of the GI tract. We have other LANCL2 ligands that have a different PK profile, that are able to be absorbed, that have high bioavailability, and those are being pursued in the context of those other autoimmune indications such as lupus, rheumatoid arthritis, multiple sclerosis, type 1 diabetes, where that systemic exposure is necessary. And they act also selectively through the LANCL2 pathway.
Gotcha. It sounds like the immune system has several layers of action. There's the general action, I guess, to take care of any attack on the body, but then there's maybe cell-specific type actions or certain pathways, I guess, that are reserved for for an immune response to certain cell types. Is that the case, or am I just speculating incorrectly?
No, I think at the high level that's accurate. I would say that the immune system is highly complex and dynamic, and it's so fluid and heterogeneous in its function that for every action, for every function that results in inflammation or destruction of a pathogen, the immune system has built in a mechanism that makes sure that that response doesn't go too far and result in destruction of tissue or of healthy tissue. So there's two arms of the immune system that are applicable to each facet of how the immune system operates. One is called the effector arm, which is generally inflammatory, destroys infected cells, destroys cancer cells, and the other is the regulatory. The LANCL2 pathway acts at both levels. It decreases excessive effector responses by decreasing TNF-alpha, interferon gamma, IL-6, MCP1, and at the same time, it enhances these regulatory anti-inflammatory responses that are critical to bring the immune system and the tissue back to homeostasis and provide, in this case, mucosal healing.
What about the microbiome associated with the areas that you're looking at? I would think that plays possibly a major role. Have you looked into that at all?
Yes, the interaction between the mucosal immune system, the immune system in the gut, and the microbiome is fascinating. So we have trillions of bacteria, and by itself, those trillions of bacteria have complex networks that interact, and then they have mechanisms of communicating with the host, with the epithelial cells, the immune cells, through metabolites, through changes in the nutrient status by changing the production of proteins by the immune system. We have looked at the effects of BT11 on the host site, and we've described that BT11 is crucial at regulating CD40 cell responses. We've looked as to whether BT11 is having a direct effect on the microbiome. And our data indicates that, like for instance, if we culture BT11 and a mixture of gut bacteria, we don't see any change in the microbiome. However, if we give BT11 to, let's say, mice, and then we measure how those mice that have been treated with BT11 have differences in microbiome when compared to mice that are not treated, we see that the treated mice have a microbiome that is richer in commensal beneficial bacteria. And so since BT11 doesn't have a direct effect in the microbiome, what we are hypothesizing is that the effects of BT11 in the microbiome are indirect, and because there's less inflammatory responses within the gastrointestinal tract, the lower inflammatory responses reshape the microbiota composition, and that reshaping results in a more beneficial commensal microbiota in treated with BT11 individuals when compared to non-treated. But again, we don't think that BT11 is having a direct effect on the microbiome. BT11 changes the immune system, and those changes in the immune system in turn result in a more balanced microbiome.
Yeah, maybe they allow a restoration of a more healthy microbiome, which therefore then positively reinforces the change that you've made. Maybe that's part of the mechanism of action.
Exactly. Once you have a more balanced microbiome with a greater presence of commensal beneficial bacteria, then those bacteria will start producing signals that will result in long-term mucosal healing. And so it's everything is part of an ecosystem where we are looking at how do we change the immune system to turn off those excessive inflammatory responses, how are we increasing the regulatory side of the immune system, and then what are the downstream implications of these therapeutic efficacy of BT11 that happens within the host site in the intestinal wall, how are those trickling down in changes, beneficial changes at the microbiome level that in turn provide that longer term restoration?
Yeah, I guess maybe it's too much to consider, but what about diet during the during the administration of this protocol? Have you modulated like the mice's diet to see how it it affects it dramatically?
No, the diet part of it is a component that is very dear to our heart. With one of the reasons is that one of our core competencies is that interface of immunity and metabolism, how the immune system changes in the context of more glucose or less glucose, whether that energy comes from oxidative phosphorylation versus glycolysis. We've determined over the years that an immune system that uses more oxidative phosphorylation as a source of energy tends to be a more regulatory, anti-inflammatory immune system, particularly CD4 T cells, whereas those glycolytic pathways are more connected to TNF-alpha production and more inflammatory responses. So I would say the underlying diet definitely plays a role in the outcomes. As you can imagine, the studies that we are conducting are quite controlled in terms of not only a protocol of treatment with the drug BT11, but having some standard diets. The interesting thing is that we came across the LANCL2 pathway, our target for BT11, in the context of studying a natural compound. We were studying abscisic acid, which is found in very small concentrations in fruits and vegetables such as figs, banana, and so on. But it's also produced in very small concentrations by immune cells in the context of inflammatory or immune stress from the carotenoid acid pathway. Of course, it's a natural compound, so the level of potency was limited. But we used that natural compound to begin to understand the LANCL2 pathway. And then we realized, so a natural compound is activating LANCL2 and it's decreasing inflammatory responses. Can we design a drug that will more potently activate that pathway and that we can use for treatment of Crohn's, UC, and other autoimmune indications? So everything goes back, to some degree, back to the diet. But of course, the BT11 program is a drug development program, even though we came across the LANCL2 pathway, actually studying a natural compound that can be found in the diet.
I don't know if you said this before. How do you induce Crohn's or IBD in mice?
Yes, there's a variety of animal models of inflammatory bowel disease in mice. We've used 5 mouse models. One of them is called the dextran sodium sulfate model, in which we use a chemical that targets the epithelial layer of the intestine, and that results in inflammation, and we've used these in mice and pigs. We've also used the interleukin-10 knockout model. In this case, the mice are genetically modified to not have interleukin-10, which is an anti-inflammatory cytokine. So in the absence of this anti-inflammatory cytokine, the mice develop a spontaneous, what's called panenteritis, which is inflammation throughout the gastrointestinal tract. It can involve the colon. It can also involve the ileum. And so we've used that model and shown efficacy. We've also used the adoptive transfer model, by which we've obtained CD4 cells from donor mice, and then we've injected them to immunologically naive recipients, and those immunologically naive recipients then allow those CD4 cells to differentiate into effector cells that produce inflammatory cytokines such as interferon gamma, TNF-alpha, and so on. Those cells migrate into the gastrointestinal tract, to the colon particularly, and in some cases the ileum, and cause inflammation. So those are 3 models, and there's 2 more we've used. A bacterial-induced model of IBD called the Citrobacter rodentium model, which is basically we infect mice with the bacteria, and then we induce intestinal inflammation. And the last one, which is very valuable from a translational perspective, is the MDR1 knockout model. In this case, there's an initial change in the ability of epithelial cells to deal with transport of metabolites that results in leakiness in the gut, and eventually there's an involvement in CD40 cells that infiltrate the gastrointestinal tract, and it becomes a very similar to Crohn's disease model. And on top of these 5 models, we've developed and used the pig model of inflammatory bowel disease, again using the dextran sodium sulfate model, the chemical that initially wipes the epithelial barrier and then induces inflammation mediated by macrophages or by T cells. And I think that something very unique about the BT11 program is that we have shown efficacy in each of the animal models that we've used. Most drugs that are currently in the market have been tested in 2 to 3 models, and they've shown efficacy maybe in 1 or 2. We have shown efficacy of BT11 in these 5 models, and in addition, we've used the pig model to better understand the PK-PD relationship within the GI tract, not only of BT11 drug substance, but the drug product, so basically the tablets that we developed for our phase 1 and phase 2 studies. So there's a broad range of models. Those are the 5 models that we've used. There's other models that we haven't used. But I think in a nutshell, those would be the top models in inflammatory bowel disease.
So you're able to induce it, at least 5 different ways. What does that tell you about how it may likely happen in people?
Yes, that's a great question, and I think that there's always the thought that whatever you discover in an animal model, you need to be able to assess the translational value, right? So that there's controversy. Well, if you see efficacy in one mouse model, does it mean that it will for sure demonstrate therapeutic efficacy in humans? Well, to address that, we haven't tested BT11 in one mouse model. We've tested it in 5 mouse models, and we've tested several LANCL2 ligands. So BT11 was part of a library. Some of the other compounds that we did not move forward had shown efficacy maybe in 2 models, but not in the others, some of them in 1 model. So we've moved forward the asset that have shown greater efficacy. And then on top of that, as I was mentioning earlier, we've gone the extra mile of choosing an animal model that is highly recognized to be closer to humans from a translational perspective. So pigs are more similar to humans in terms of gastrointestinal tract anatomy, physiology, immunology than mice. And so we wanted, as an intermediate step before moving to humans, to have some of that pig data. And not only we've seen efficacy in the 5 mouse models, but the pig model also demonstrated efficacy. And then there's some biomarkers that we've seen consistently changed in the context of BT11 treatment. One of them is fecal calprotectin. Fecal calprotectin is produced by neutrophils. It's an inflammatory cytokine. It's considered a biochemical measurement of inflammation in stool. And elevated fecal calprotectin levels indicates the migration of neutrophils, which are inflammatory cells of the immune system, into the intestinal mucosa. We have seen across these animal models that BT11 can decrease calprotectin in stools, and moreover, we've shown that the ability of BT11 to decrease fecal calprotectin outperforms that of other drugs, such as TNF-alpha blockers or 5-ASA. So a key question that we asked in our phase 1 clinical trial was, is BT11 capable of decreasing fecal calprotectin? Will we be able to use this biomarker? And our data, and we published this in Inflammatory Bowel Disease Journal, show that yes, not only BT11 is safe, as I mentioned earlier, even tested at high doses such as 100 milligrams per kilo, which is about 7 grams of BT11, we saw no differences in adverse event profile when compared to placebo. But more importantly, we saw that drop in fecal calprotectin. So there's some indications of translation along the way. Does it move from mice to pigs? Do we still see the efficacy? The answer was yes. Do we see the changes in fecal calprotectin in humans? The answer is yes. And so does it mean that the mouse models are infallible? No. In fact, in many cases, they have not translated. But if you use the mouse models in combination of, translational assays, carefully selected biomarkers, and then go the extra mile on choosing a large animal model that is more similar to humans, the likelihood that those predictions will be accurate also increases.
Wow, all right. You looked at this from many, many angles so far. So what's the pathway from here? You said that are you through phase 1 clinical trials, or what's the pathway until this will actually be in use?
Yeah, so we completed phase 1 clinical testing in December of last year. We've initiated phase 2 study with 195 ulcerative colitis patients. We have 3 groups, low dose BT11, high dose BT11, placebo. The study will have a 12-week induction followed by a maintenance phase, and we'll have endoscopy at baseline, 12 weeks, and at the end of the maintenance phase. And so we'll have the induction phase data in the second half of 2020, so next year. Once we have that data, we can talk with the FDA, have an end of phase 2 meeting, begin planning and implementing the phase 3 study, which will be a larger ulcerative colitis study. And the drug could be in the market as early as 2023. So depending, of course, on the results of the phase 2 and phase 3 study. So now we are focusing on implementing this phase 2 study. We'll be starting a Crohn's disease study next year with about 130 Crohn's disease patients. And we'll also be advancing some other assets that are currently in the pipeline in IND enabling stage to phase 1. So we are very excited not only about BT11, but other assets in our pipeline.
That's great. So how can people find out more and keep pace with what's going on and when as the drug moves through the clinical trials?
So we are updating our website on a regular basis. They can visit our website at landosbiopharma.com. And we will also be having some press releases over the next few months announcing the progress that we are making with the interim analysis phase 2 results. We are engaging additional sites, and we are very excited about our ability to help address this unmet clinical need and ultimately serve the patients with autoimmune disease with BT11 and other drugs in our pipeline.
Very good. Well, Josep, I appreciate you coming on the call. Thank you for being here and for your work.
Thank you very much. Thanks for having us.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.